Auzubillahiminashaitanirrajim. Bismillahirrahmanirrahim. In the name of Allah, the most merciful and beneficial. My presentation topic is fermentation industries of Danish, submitted to Dr. Hamad Majid. The subject is industrial chemistry, submitted by Hadija Tariq, roll number 2. First of all, I will discuss the table of contents of fermentation industries of Danish. Number first, top industry fermentation industries. Number second is Novozyme. Number third is industrial setup of Novozyme. Number fourth is Core. It is also an industry of Danish. Number fifth is industrial setup, and at last I will be discuss about the references of this all subject of this all topic. First of all, top fermentation industries. The Danish biotech and Druid genetics has shown it can improve the yield of fermentation by engineering bacteria to become addicted to being productive. It means that we improve the fermentation yield by using the engineering bacteria to improve our yield of the quality of the product. Next one is fermentation was full of bacteria used or the workhouses for industries such as the pharmaceutical and in chemical industry. They are used to mass produce products including detergents, drugs, and biofuels. The fermented products are used to form in detergents and drugs and fossil fuels. Best fermentation industries are the top best are Novozyme and Janico. These both are the fermentation industries of Danish. I will be discuss these two industries one by one with industrial setup. Number one is Novozyme. Novozyme is a global biotechnology company headquartered in outside of Copenhagen, Denmark. The company's focus is the research, development, and production of industrial enzymes, microorganisms, and biopharmaceutical ingredients. It is the world best Danish company of fermentation. In this company, we will form the enzymes, and these are used in many in purposes. In Novozyme company, we use the submerged fermentation and other. In submerged fermentation, we will know that cells are cells are um, present in the free in fermental, not not move cell easily moved in it. This is called submerged fermentation. In this industrial setup, we will see the and that at first raw material is added. The mixing of raw material is take place. Continuous sterilization means sterilization take place to form the aseptic environment. Then it is added to the feeding acidify and antifoam. Then the lyophil viral is added to agar medium. It is the formation. These are used to the formation of inoculum. In previous slide, yeah, or previous lecture, I will be discuss what is inoculum. Inoculum is pure and healthy. It is a cell form which are used to form the yield of the fermentation. It is pure contamination free. So the sterilization of inoculum should be done. Filtration should be done. Air is added in it. Air is added in the fermental in also the in inoculum tank by using by air. We use air for from the environment. So at first we sterilize the air, and then we will add it to the inoculum tank or and the fermenter tank. Then the in fermental tank the submerged fermentation take place. Water pressure, mass, volume, temperature, pH, dissolve oxygen, enzyme activity, biomass, etc. We will be all these are things measured and monitored. With these all the things measured and monitored, we will form the yield of fermental best and improve yield. And we also measure the carbon dioxide amount, oxygen amount, airflow, etc. in gaseous exhaust. In next slide, in this slide, I will be discussed about Genicode. Genicode is the other industry of the uh, uh, Danish. It is using fermentation technology. Genicode scientists design protein for customers at the FIM Rochester NY laboratories. In this laboratory, Rochester's laboratory, scientists design proteins which are profitable or which are important for the customers. With the Danisco acquisition of US firm Janico, Danish companies now control two-thirds because of the presence of Janicore. Two-thirds of the global enzyme production take place. This is the industrial setup without the Janicore and with the presence of Janicore. At first industrial setup, the conventional dry mill ethanol production process. In this process, first grinding, then slurry, then the temperature is needed in the liquefaction. First primary liquefaction, secondary liquefaction, then saccharification, then fermentation, at last distillation and dehydration. And this is the recovery of the alcohol, then storage tank. But in the presence of genic core strategy, we will skip the liquefaction step and combine the fermentation and saccharification step in one process, and the temperature amount is not changing in it due to the presence of strogen. At Genicore industry, first the formation of auxiliaries enzyme. This enzyme is used for the production of ethanol in large scale, large amount, and it should be improve the fermentation yield. Genicore strategy is very important and is very useful. It is maintain the temperature. We should not to decrease or increase the temperature. We should not done the liquefaction step. We should not the liquefaction step at specific temperature. We done it at any temperature with fermentation, with the combination of fermentation. That's why Genicore company is best and one third enzyme production company. At last, these are the references. The Novozyme report 2021, Novozyme 1 1st February 2022, and Novozyme report 2014, Novozymes 20 January 2015, and Denisco acquired Genicore, Genicore 2010. At last, thank you so much. Any questions? That is from my side.